Welcome everybody, this is Dr. Jenkins, lab number 2 on osmosis and diffusion. So this is a lab that I think having a video to really talk through it will help you, okay? So I'm glad you're listening. Fight the urge to fall asleep or just hate it. Stay with me because this is gonna help you understand the material, do well on the lab assignment, and do well on the lab quiz, all right? So here's, see if I can figure this out. Let's see, there we go. Here's the outline, and I know you might not be smiling. Let's see, what color can I use today? Let's use purple, okay. I know you may not be smiling like this guy, and you're probably thinking, osmosis and diffusion. Ugh, I've been hearing these words since I've been in 5th grade, da, 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 da. But we are gonna talk about it and we're gonna get you through it. We're gonna talk about why it's important. And then once you do well on your lab assignment and lab quiz, you, my friends, are going to be smiling. Okay. All right, so make sure that we understand some basic definitions. We know that every cell, it could be a nerve cell, it could be a muscle cell, any old cell, has a specialized outer membrane, and that is called the plasma membrane. And if we magnify that plasma membrane, you'll see, and you've gotten this in a lecture, so you already know, I'm just reviewing it because the more times the better. When we magnify just this outer layer, it is a phospholipid bilayer. So we have 2 layers of these little phospholipids, right? The heads face in and out, the tails face in. And there's some things interspersed in between. But the idea of this is that it is selectively permeable. So it will allow some things to diffuse in, but not others. When I'm talking about this in my lecture, I talk about the selective permeability like the bouncer at a club, you know? The bouncer at a club can let in the pretty girls or can let in the people that slip them some money, but the bouncer at the club is selectively permeable. Doesn't allow everything in, but is selective. And that's good for the cell. I mean, you know, it's easy to talk about the nucleus and all the other fun stuff inside the cell. But if we don't have a proper boundary, that means any old thing could come in, and we don't want bad things that come into the cell because that cell will die. So the outer plasma membrane is really important, and the fact that it is selectively permeable is also really important. We need to bring in oxygen, we need to bring in fuel like glucose, all sorts of fun stuff. So when we talk about getting things through that plasma membrane, there are 2 ways. We can move something through the plasma membrane passively, which means no energy is required. And that's what we're going to talk about today, because diffusion and osmosis are passive means of transport. Or sometimes we have to push things through active transport. And this requires energy in the form of ATP. And this little picture on the right does a really good job of explaining this. When we have passive transport, things are able to move because they move from a high concentration gradient to a low concentration gradient. Therefore, it does not require any energy. When we're talking about active transport, you are trying to go against the grain. You're trying to pedal up a hill. You're trying to row a boat upstream. So because we're going against the concentration gradient, it requires energy. See, I told you, this is good stuff, right? Yes. All right, so we're focusing on passive transport. into and out of the cell, and we're going to be talking about diffusion and osmosis. Make sure you review these definitions. Diffusion is movement of a particle from high to low concentration. Movement of a particle from high to low concentration. My cat is here scratching. Hey, my cat is scratching on my seat here. I'm not going to have that in Dr. Jenkins's house. And then osmosis is the same principle. The only difference is that we're talking about water. So you could define osmosis as diffusion of water, or you could simply say movement of water from high to low concentration. So they're really the same things. It's just that when we're talking about diffusion, we're referring to a particle not named water. And when we're talking about osmosis, we're talking about water. And we could see how this could happen. So let's say outside of the cell, because this is outside of the cell, and this is inside of the cell. If I have a lot of water, let's draw in some water molecules. If I have 1, 2, 3, 4 water molecules on the outside and only 1 water molecule on the inside, well, guess what? There's a higher concentration gradient outside and a lower concentration gradient on the inside. So water will diffuse from high to low. In order to have osmosis and diffusion, we have to have that selectively permeable membrane. So we have to have little gaps in between those phospholipids and or channels. You can't diffuse through a solid membrane. It's got to be at least selectively permeable. And you must have a concentration gradient. Perfect. See? Not so bad. Now, why do we care about this, folks? We care because pretty much everything in our body is founded or involves these principles. So I already mentioned a cell. If we have, let's even take an example like a nerve cell. We have a nerve cell, there's the cell body, there's the axon. Well, cells, they need things to operate. We've got to bring in oxygen. We've got to bring in glucose. It's a really bad glucose molecule. We also have to be able to get rid of waste. Cells are living, breathing, operating things, so they produce waste like CO2. So we have to be able to bring things in and push things out of the cell. And it's not just that. There's a lot of body processes that are founded in diffusion. How do we exchange gases in our lungs? Diffusion. How do our kidneys filter blood? Diffusion. So it's something so simple. It's almost like its simplicity is brilliant. That the body sets up things to have concentration gradients between a selectively permeable membrane and therefore can utilize diffusion, which is great because it doesn't take energy and it happens. All right, are you happy yet? You might be happy now, but before we get to the lab, I'll ask again at the end. All right, so going through this lab procedure, number 1 is to define and look at Brownian motion. Brownian motion is simply that particles have this natural kinetic energy. They're jumpy, they're jittery. So this picture is trying to show you these little purple molecules, they have a natural random kinetic energy. You know, sometimes the kinetic energy of a particle will push it this way, sometimes it'll push it that way. But the idea is that particles are naturally, randomly jumpy. And this, my friends, helps diffusion. Because if particles have this natural kinetic energy, it's going to help them be more likely to diffuse from high to low concentrations. Diffusion is still passive, but the fact that molecules have this natural energy It just helps diffusion along. I'm going to post this link for you also, where you can just pause the video and you can copy it here. This is a quick little video that shows you the particles just naturally moving. Okay, pretty simple. Now we get to the meat and potatoes of this lab. So understanding and being able to define osmosis and diffusion is one thing, but we're going to take it to the next step because we want to, in order to better understand osmosis and diffusion, we're going to talk about what are some things that influence it. And in this lab, we have 3 different experiments that we usually would be doing in the lab, but here we're going to be talking through it virtually. We're going to be looking at how concentration gradient, particle size, and particle weight influence osmosis and diffusion. Let's first start with concentration gradient. All right, now, if we were in the lab, we would be doing this. I'm just going to walk you through it. Just took a sip of water there. Okay, so in this lab, you would usually be working with dialysis tubing. When it's dry, it looks like this and it's kind of crinkly. But when you submerge it in water, that dialysis tubing becomes pliable. And we're using this because even though you can't see it, there's tiny pores in this dialysis tubing, which means that, yep, you got it. I knew you were with me. That membrane is selectively permeable. And actually it's called dialysis tubing because it is what is used in a dialysis machine. So people whose kidneys are not able to function go and get dialysis, and the dialysis machine basically acts as their kidneys. So if you're not familiar, in a dialysis treatment, the person will have blood taken out of their body through an IV needle and through little tubes into the dialysis unit, and that dialysis unit has a bunch of this tubing which allows for the unit to filter the blood. So this is selectively permeable, so it can filter good things in and bad things out. All right, anyway, so you would be taking these pieces of dialysis tubing, you'd be tying them at one end, and then we're going to be filling it with a sugar solution. Sucrose is sugar. And then after you fill it with a certain amount of a sugar solution, you tie the other end. So what you would be left with are 3 bags that look like this. They're dialysis tubing bags. They are tied at either end, and inside is a little bit of sugar solution. But because we're looking at concentration gradient, we have 3 different concentrations of sugar. Okay? In our first tubing bag, it's a 25% sugar solution. And I want to draw this out a little bit because in order for us to determine what's going to diffuse and in what direction, you simply have to identify what the concentrations are of the fluid. And this is going to make sense more in a minute, bear with me. But let's take this first bag. If it is filled with a 25% sugar solution, that means that the rest of it is water. So you would, in the lab, you'd have a little dropper, like an eyedropper type thing. And in that eyedropper would be a 25% sugar solution. So it'd be 75% water with 25% sugar. Think about a really weak, watery Kool-Aid. The next one would be 50% sugar and 50% water. So that would be like a stronger, more sugary Kool-Aid. And then the last one is even sugarier, if that's even a word. So it'd be 75% sugar and 25% water. All right, good. So we've laid the foundation of our 3 different concentration solutions. What we would do is we would put these bags on our scale and we'd weigh them initially. So you can see I filled in, because I did this lab myself just for you, we filled in the initial weight. And what we're going to do is we're going to submerge these bags in water, and then we're going to reweigh them at 20 minutes and 40 minutes. So it doesn't necessarily matter what the original weights were. What matters is going to be our change in weight. Excellent. So now we put each each bag into a beaker of water. And just to make sure we look at water, just imagine there's water. Boop, boop, boop. Okay, so we submerge these bags, and remember, one of them was a 25% sugar, one was a 50% sugar, and one was a 75% sugar. And we're going to submerge them each in a beaker full of 100% water. Now, this is where you, because you'll need this information for your lab assignment, I want you to create a hypothesis. Will the dialysis tubing bags gain weight or lose weight? And if they do, which bag is going to change the most? I will pause. Okay. Now, I couldn't just leave you hanging there without giving you a little bit more help. So let me explain more of what I was saying before. That if we're trying to determine osmosis and diffusion, the easiest way to determine what's going to happen is to simply lay out the concentration inside versus outside. In this case, we have a dialysis tubing bag, right? And that dialysis tubing bag is submerged in water. And we're going to have the same thing for all three, where we have a dialysis tubing bag, and it's going to be submerged in 100% water. So if we want to determine how the sugar is going to diffuse and how the water is going to diffuse, all we have to do is lay out the concentrations inside and outside of the bag. So it's really, once you lay it out, I don't think it's as difficult as maybe it might seem to you at first. So let's take our bag number one. And our bag number one had 25% sugar and 75% water. Bag number two had 50% sugar, and then the remaining 50% was water. And then, of course, our bag number three had 75% sugar, which meant 25% water. So those are our solutions inside the bag. And then what is our solution outside the bag? Well, in the beaker, it is all water. So that, my friends, is easy. This might seem a little tedious, but once you lay it out, then we can clearly see what's going to diffuse in what direction. Let's look at, we'll use green. Let's look at the sugar. Let's look at bag number one. Let's compare the sugar. We have 25% sugar here and 0% sugar here. So we have a higher concentration of sugar inside the bag and a lower concentration of sugar outside the bag. So how will the sugar diffuse? From inside the bag to out. Right? So that's going to be our movement of sugar. Moves from high to low. But we are going to be looking at the water. Yes, sugar will diffuse. In this case, it'll diffuse from inside the dialysis tubing bag to the outside. But because it's such a small amount of sugar, it's not going to influence the overall weight of the bag. And this is going to be true for the semester moving forward and even in the body, right? And we're talking about what's going to really cause the most change in weight or the most movement, we're going to be looking at the water. And just so we can better see it, let me use orange. So in order to see about the weight change of the bag, we're going to be looking at the water. There's more water, therefore it is the movement of that is going to potentially change the weight of the bag. So now let's look at in bag number 1, let's look at the water. Outside in the beaker, we have 100% water. Inside, we have 75% water. So water, just like everything else, is going to move from high to low. So in this case, we have more water outside the bag and less water inside the bag. So water will diffuse in. And if water diffuses into that bag, what do you think is going to happen to the weight of that dialysis tubing bag? Yeah, it's going to gain weight. All right, so I've outlined how you look at this, and I've given you an example for bag number 1. That bag will gain weight because water will diffuse from higher concentrations outside the bag to lower concentrations inside the bag. I'm going to let you do the same thing for the remaining 2. Okay, so you're going to compare, let me go back to orange. You're going to compare this to this and this to this and tell me for bags number 2 and 3, will they gain weight? But the other question is, which bag might have a weight change that is the greatest? Because we're looking at concentration gradient. In all 3 of these, there's a concentration gradient. There is, but we're looking at the degree to which there's a concentration gradient. If there's a larger gradient, does that mean that there's more diffusion? You're going to find out, my friends. Feel free to pause the video if you need. And I've gone ahead and I'm going to give you some of the answers here. So at 20 minutes, we'll keep with orange. At 20 minutes, we can see our weights. So you can ask yourself, out of these 3 bags, which one gained the most weight? Perfect. Now we're going to wait another 20 minutes, so it'll be a total of 40 minutes. And here's my next hypothesis question for you. If we have time 0, 20 minutes, and 40 minutes, we already saw what happened between time 0 and 20 minutes. Do you think that the same trend will continue from 20 to 40, maybe? Or maybe there won't be any more diffusion? And I'm going to give you a hint to think about this. As time goes on, what do you think happens to that concentration gradient? If it started off as being a really high concentration gradient, after 20 minutes, once things have begun to diffuse, the concentration gradient will be lower because things will have started to equal out on both sides of the bag. So what does that mean in terms of the amount of diffusion that is going to continue to happen for the next 20 minutes? See? Good questions, right, folks? Yeah, I told you. All right, well, here we go. Here's your answer. And I'm going to let you figure it out, but here's the data. So you'll be able to compare the weight change from time 0 to 20 minutes for all 3 bags. And then you'll also be able to compare the weight change from 20 to 40. So you're going to have to answer this in your lab assignment. What happened to the rate of diffusion in these 2 time blocks? And of course, the answer would be based off of the degree to which there was a concentration gradient. The last thing you'll do is you're going to calculate the total weight change. So you would take the highest weight out of all 3 times, highest weight minus the lowest weight. Excellent, my friends. Now you'll be able to answer the questions in your lab assignment. Good stuff. Okay, so we've talked about concentration gradient, how that affects diffusion. Now let's look at particle size. Pay attention here, we're not necessarily talking about weight. This next experiment is talking about size. So in this lab experiment, we're looking at 3 different particles: glucose, sodium chloride, and starch. And I've put these pictures a little bit to scale so you can see some of the smaller molecules versus a bigger molecule. So my question to you is, do you think it's going to be easier for a smaller or a larger size molecule to diffuse across a selectively permeable membrane? Here is how the experiment is set up. We're again working with the dialysis tubing, and we would start with a bag, tie one side. Only this time, we're putting a combination solution. So we're filling this dialysis tubing bag with a solution that contains starch, sugar, and sodium chloride, all 3. Then we're going to tie the other end of the bag, we're going to weigh it, and then we're going to submerge it into a beaker full of water. We're going to wait 40 minutes, take the bag out, reweigh the bag, and then we're going to examine the water contents from the beaker. Okay, so here are the hypothesis questions for you. And again, all you have to do is lay it out. We have a dialysis tubing bag on the inside, and it has a combined solution of sugar, salt, starch, and of course, on the outside of it is water. Now, in this one, it didn't give you an exact percentage of the sugar, salt, starch solution, but it's probably somewhere about 3% sugar, salt, starch solution. So if it's 3% sugar, salt, starch, that means it is 97% water. So with this information, you're going to be able to figure out, number 1, is this bag going to gain weight, lose weight, or stay the same weight? And then number 2, out of these particles, sugar, salt, and starch, will any of them diffuse out of the bag? Now, in this question, we have to consider concentration gradient. So there has to be a concentration gradient present, but we're also considering size. So remember, the dialysis tubing is only selectively permeable. It has pores that are only a certain size. So out of these 3, sugar, salt, starch, out of those 3, which one or which ones do you think are going to be small enough to fit through the pores of the dialysis tubing bag? Great, make sure you write those down before we get the answer. Okay, so I've given you the results for the weight. We have our initial weight. We have the 40-minute weight of the bag, and you can see what the weight change was, and then you're going to figure out the total weight change. Pretty simple. And then, here's what we did. We took the bag out, and then we tested the beaker water. So we test this! Exclamation point for all 3. And what that means is whatever of the sugar, salt, starch, whatever ones were able to diffuse out of the bag and into the water, then the water would test positive for. And here are our answers. The beaker water tested positive for glucose and sodium chloride. So this is for you to answer in your lab assignment sheet. Out of the 3 molecules, sugar, salt, and starch, which of the 3 were small enough to diffuse out of the bag? Were there any that were too large and therefore not able to diffuse out of the bag. We may have a concentration gradient, but if a particle is too big, it simply may not be able to fit through the pore. Life-altering material here, isn't it? Yes, my friends. Okay, you're going to answer the questions in your lab assignment. Got it. Now we're going to be looking at our last thing that can influence osmosis and diffusion in this lab, and that is particle weight. We previously looked at particle size, and now we're looking at particle weight. Now, I want to make a point here. When we look at weight, this is independent of size. So I think people commonly link these together, that if something is a heavy weight, it's probably a large size. And while sometimes that may be true, it isn't always true. For example, a balloon, when you blow it up, a balloon can be large in size, but lightweight. So try and take size out of the equation for this one, out of your thought process, and think only about weight. So the question is, does a lighter weight or a heavier weight particle diffuse more? Here's how the experiment works. We've got 3 different weight particles, and we're going to put them into a little agar dish. Some call it auger. I say agar, whatever. It's basically like that little jello-like substance. So we poked a hole, and then we put 1 drop of these dyes, and we wait an hour. So here we can see our 3 different dyes. So we just put a drop of dye. One is blue, one is green, one is purple, the potassium permanganate, even though here it kind of shows the potassium permanganate is orange. And just by the nature of the dye, they are different weights. So my question to you is, out of these 3 weights, now that we can see which of the dyes are a heavier weight and which are a lighter weight, which will diffuse more, which will diffuse the most through the agar? And remember, this is independent of size. I know, I can't think of the word. You're just so excited you can't wait to get to the answer. I know, the suspense, that's the word. The suspense is killing you. Well, for this one, I'm going to let you do the measuring, folks. So we can see in this picture, the dyes have diffused out. In this first picture, they were all small because at the beginning, we just put 1 drop for each. But here we waited an hour and they diffused. And I'm going to let you do the measuring because I've put up a ruler. We're going to use millimeters, okay? So you're going to be able to do it. So I'm going to let you... Oh, for some reason, all right. For some reason in this video, these are flipped around to the wrong way. Okay, so look, we're all trying to get this online thing and purpose. So instead of using millimeters, because those are the wrong way, we're going to use inches. And I hate to do that as a scientist, but we've come this far. So this side of the ruler is inches, and this side is centimeters and millimeters. So you're going to have to do this on your own. You can pause the video. And from here to here is 1 inch. Right? And this would be 14.5. So you're just going to have to count, right? So you want to count from this side to this side and count how many inches or how many parts of an inch it would be, okay? Each one of these lines, by the way, is 0.05. All right, you are smart enough to figure that out. Then you're going to do the same thing for the blue, okay? So you're going to get some numbers that'll look like, I don't know, 0.4 of an inch or something like that, okay? And then you can do the last one here. I've lined up the green. So don't tell the science guys that we're using inches this time. Just a mistake on my part. Okay. So, I mean, even if you go back and look with your naked eye, you can see which color diffused the most and which color diffused the least. And the point here is to compare which one diffused the most and least with the particle weights. So you'll see a trend. Does a lighter one diffuse more or a heavier one diffuse more? And in your lab assignment, I'm going to ask you to do some measuring here. Doesn't have to be exact, just give me an idea. And then you'll see the answer. All right. Okay, we are coming down the home stretch here. Stay with me because you are going to benefit from this discussion. I promise you that. All right, the last part of this lab is a different but related property called tonicity. Are you ready? All right. And I'm going to give you a definition that works for me. I think the textbook definitions are a little complicated for no reason. So when I think of tonicity, I really think of it as how a cell might change shape depending on what diffuses into or out of it. So tonicity is this characteristic where a cell, here we have a red blood cell. Tonicity is a characteristic where a cell may change shape. For example, if you have a bunch of water and all of a sudden a ton of water diffuses into that red blood cell, if water rushes into a red blood cell, wouldn't the red blood cell get a little bigger? It'll change shape, absolutely. And red blood cells are a good example here because they are flexible and flimsy, so they're able to change shape. On the other hand, what if all of a sudden a lot of water diffuses out of a cell, a red blood cell? It just makes sense. If we have a red blood cell that all of a sudden loses a bunch of water, that red blood cell may get smaller. And this characteristic of how a red blood cell may change shape is called tonicity. Because keep in mind, folks, diffusion is happening all the time. Things will diffuse into the cell, things will diffuse out of the cell. And as a result, depending on how much, and it's mostly we're looking at water here, depending on how much water diffuses in or out, the cell may change shape. That's not complicated now, is it? Now, of course, people in the science world, they want to try and make it complicated. So we have names for these types of solutions where a cell may get bigger, a cell may get smaller, or a cell will stay the same shape. And I can see in my formatting and coming over to my iPad here with my fancy pen, we've covered out the isotonic, but no worries, we're gonna still go on here. When you look at the textbook definition for these tonicity solutions, it's a little bit confusing. So I encourage you to go with my definitions to start with. Later, if you want to look at the textbook, I can explain that to you. But I think the easiest way is just to keep it simple. Let's start with hypertonic. A hypertonic solution is any solution in which submerged red blood cells get smaller. Hypertonic. You can see in this picture, this red blood cell is losing water, so that red blood cell gets smaller. A hypotonic solution is the opposite. Hypotonic solution is any time when a red blood cell is submerged in that solution, that the red blood cell takes on water and gets bigger. Which leaves us isotonic. And isotonic is any solution where the cell shape stays the same. Okay, let's unpack this a little bit. If you look at the prefixes, let's go back to green. I like the green with this one. Hyper. We know that hyper means more. So why is it that in a hypertonic solution, the cell actually shrinks? And it has to do with the movement of water and the difference in concentrations and then the solutes as well. Because in addition to water diffusing out, there's also going to be movement of sodium chloride. You can't even read that. What we're going to find out in a minute is that inside of that red blood cell, it is 0.9% sodium chloride, and then the rest is water. So because of this, there's going to be some movement of salt. And the hyper prefix is referring to the salt concentration. But I find that to be confusing, because even though there will be a little bit of sodium chloride diffusing, it is the water that is going to be what causes the cell to change shape. It is the movement of water, not the sodium chloride. It is the movement of water that is what causes these red blood cells to change shape. So that's why I think it's easier to look at the water, even though these are named based off of the sodium chloride solutions. Now, how I remember these solutions is, I think about hyper, and don't laugh at me. If someone is hyper, they may go see a shrink or a psychiatrist. So that's how I've remembered for years and years and years, that it is a hypertonic solution that the cell shrinks. Anytime you can utilize, even if it seems stupid, anytime you can utilize some little thing to remember it by, go for it. Make sure you also note here we have fancy words. So crenate is a fancy word for shrink. And then check out, in a hypotonic solution, the cell gets bigger, and the cell will frequently take on so much water, get big so quickly it can burst. And the fancy word for a cell bursting, because it takes on too much water, is hemolyze. All right, that's just memorization, folks. Okay, so in this lab procedure, if we were in the lab, this is how we would do it. We would set up these 3 test tubes. And the 3 test tubes, each would be filled with a different solution. So in test tube A, we've got a 3% sodium chloride solution. In test tube B, a 0.9% sodium chloride. In test tube C, 100% water. And as I did before, guess what? If we're going to try and figure out how water is going to move, we compare the concentrations inside and outside. So let's just start. If test tube A is filled with a 3% salt solution, that means that the rest of that is water. If test tube B is filled with 0.9, the rest is water. And test tube C is all water. Now these percentages may seem a little random to you, but they're not. So when we're talking about these, 3% is similar to seawater. So if you go to the ocean and it's salt water, it's usually 3 to 3.5% salt. And B, this is the same as saline. So an IV bag that someone gets in the hospital is saline. The solution that you keep your contacts in or wash out your eyes with is saline, and that just means it's a little touch salty, 0.9% sodium chloride, and the rest water. All right, and then to each of these test tubes, we are going to drop in red blood cells. So my question to you is, in which of these test tubes will the red blood cells take on water, get bigger, hemolyze, burst? In which of these situations will the red blood cells get smaller? And in which will it be isotonic, where the cells stay the same? Let's think about it. I'm going to go through one example, and then you'll be able to do the rest on your own. So remember, we already started this. But we can do it again. If test tube A is 3% sodium chloride, 97% water, 99.1% water, and then all water. I will tell you something. Once we add the red blood cells, there is some fluid in red blood cells. All cells have a little fluid on the inside. And we happen to know, it's just a known, what the fluid inside red blood cells is. So for all three of these, that red blood cell is a little bit salty. So it has 0.9% sodium chloride, which means the remaining is 99.1% water. And that's going to be the same for all red blood cells. 99.1% water. And we're almost done, folks. Stay with me here. Excellent. Let's take test tube A as our example. Bright green. Oh, yes. Okay. So we're going to compare the concentration outside the red blood cells to the concentration inside. Because remember, we submerged these in here. So we've got our red blood cells submerged in this case, a salty solution. Remember, we want to look at the water. We want to look at the water. So outside of the red blood cells, we have 97% water. Inside the red blood cells, we have 99% water, 99.1. So if I have 99.1% here and 97% outside, how's the water going to move? It's higher inside the red blood cell. So water is going to go from 99.1% to 97%. It's not a big difference, but water will diffuse out of the red blood cell. So you fill in the rest. If water diffuses out of the red blood cell, what's going to happen to the shape of that red blood cell as it loses water? And then consequently, what type of solution would this be called? Now, just as a quick note, salt would diffuse the other way because there's more salt outside and less salt inside. So sodium chloride will diffuse in. So that's happening, but we are going to look at the water to see how the shape of the cell might change because there's more water. You do the rest, my friends. Excellent. You're going to be able to answer the lab questions. I'm going to post a video of what it looks like when these cells take on water or lose water. And I'm going to leave you with a couple things here. So I mentioned, let's see, was there orange? Good. I mentioned seawater and saline. Perfect. Once you complete this lab and you have labeled which test tube would be a hypertonic, hypotonic, and isotonic solution, you're going to be able to understand my next question. And we actually already answered test tube A, didn't we? Because test tube A was going to see water. Did I have this right? Let's just make sure I know what I'm doing. Yeah, test tube A is going to see water leave the cell, right? So water is going to leave the cell, and I'll just give you this answer. That means that the cell is going to crenate or shrink, and this is a hypertonic solution where the cells get smaller. Now, folks, if our red blood cells lose water, a lot of water, and get smaller, do you think that those red blood cells can continue to function normally? No. When these red blood cells shrink, they can't do their job anymore. So this is a dangerous situation. Dangerous. If your red blood cells start to shrink, they can't do their job, they can't carry oxygen, your tissues are not going to get oxygen, you could die. And I bring this up because, if you're stranded on a desert island, you should not drink the salt water. Why should you not drink the salt water? Because if you drank so much salt water, it's different if you're just swimming at the beach and you accidentally take a little gulp. But if you're drinking excessive amounts of that salt water, what's going to happen to your red blood cells? They're going to shrink. And your red blood cells will no longer be able to work, and you could die. One of your lab questions is going to ask you about an IV solution. So if someone goes into the hospital because, say, they're dehydrated, would you want that IV solution, that IV bag, to be 100% water? Would you want that IV bag to be saline? Would you want that IV bag to be salt water, like a sea salt water? Understanding this will give you that answer. Hopefully some light bulbs went off in your head. Thanks for listening. Watching this video will absolutely help you. If you have any questions, email me or I'll see you on Zoom.